Thanks for this opportunity to present my work. I'll talk about whole embryo spatial transcriptomics, how we put fate into form. Embryonic development is a process of building complexity in space and time. Here you see a zebrafish embryo developing outside its mother for the first 24 hours of life. Cells go through complex patterns of divisions and migrations, which are variable in both space and time, yet tissues and organs emerge very robustly at the right time with the right timing. So, what rules do cells have to follow to turn this spatial temporally variable information into robust tissue outcome has been the focus of my study. So over the decades, many pieces of the puzzles have been identified. For example, we know that signaling pathways set up the initial asymmetries and its transcription factors that define the identity of the tissues. For example, here I'm showing you some transcription factors expressed at a margin of the embryonic gastrula stage and that define the later mesoderm and endoderm. Of course, these are not static snapshot. Everything is put into complex patterns of morphogenetic movement. For example, if you follow the dorsal margin, you know that they involute during gastrulation and go through convergence and extension to form the dorsal axis. But our understanding of this process is a little bit anecdotal in the sense that the studies are mostly focused on a single gene or single phenotype. We still yet lack a systematic understanding of how different genes coordinate with each other to form this complexity and how this coordination is put into this dynamic motion. So modern technologies has provided us avenues towards a global view. For example, single-cell RNA-seq that has allowed us to characterize dissociated cells across embryos across developmental time points, and we can use their single-cell transcriptome to reconstruct these molecular trajectories that reflect um, the fate decision process. But because these cells are dissociated, we lack the critical information where they come from and how they uh, interact with each other. Similarly, in total imaging has given us unprecedented resolution into cell behaviors. But as you can see, the throughput of genes you can visualize from this method is rather limited. So technologies that put fate into form is very much in needed. So we really need ways to critical questions remain unsolved. For example, what is the full diversity of gene expressions? How do gene patterns change with cell dynamics? And how do tissue boundaries robustly form? So to answer this question, we really need a technique that is capable of profiling many, many genes in the intact embryos. But the first challenge we face is a technical one. Traditional methods like fluorescent in situ hybridization can characterize gene expression in whole embryos, but its lack of throughput because the number of genes you can probe scales with the number of fluorophores you can put into the system, usually no more than 3 to 5. With the advancement of spatial omics technologies, it becomes increasingly feasible to profile genes in large scale. But as far as we know, all of the commercially available methods rely on sectioning the samples into thin slices, which sacrifice the coverage and makes the data very hard to reconstruct in 3D. So we decided to come up with our own way to profile gene expression at high resolution in intact embryos. We call the method whole embryo merfish or WeMerfish. This is how the method works. So we know cells have RNAs in them. We design DNA probes against these RNA molecules with acridite modifications. Um, so we can cast an acrylamide gel that cross-link very stably to these DNA probes. Then we can clear away all the lipids and proteins in the system so it becomes very transparent. We mount the sample onto a spinning disk confocal microscope coupled with a microfluidic system so we can flow in the linker probes and fluorescent readout probes. We can either measure one gene expression pattern per round, or here we use the combinatorial approach called Merfish, where instead of a single gene, we image the different combination of different genes, and based on single molecule colocalizations, we can decode which transcript belongs to which gene. So from the molecular trajectories I mentioned at the beginning, we identified 495 genes that are spatially or temporally variable and designed a library against it. Furthermore, I developed a branching signal amplification approach that boosted the signal by a factor of 3 to combat for the signal loss in confocal imaging. Through these 4 points of technological innovation, we were able to achieve high resolution gene profiling in intact embryos. This is what the data looks like. Take the 6 somite stage embryo as an example. Here's an overview of the embryo. If I zoom into a single field of view, you can see the tissue architecture is largely preserved. Here's an XZ or cross-section of the sample where the scale bar is 10 microns. You can see we can image hundreds of microns into the tissue without losing signal. If I further zoom in, here I'm showing you 2 genes imaged sequentially, MYOD1 in the adexal cell and TBXTA in the notochord. I hope you can appreciate the single molecule signal as well as the sharp boundary between these 2 tissues. Additionally, we have oligoconjugated antibodies targeting the membrane, so we can carefully delineate the boundary between cells, and we can assign these single transcripts into individual cells or even subcellular compartments. So here's a collection of the 495 genes that we imaged. So we have 3 developmental stages: early gastrula, mid gastrula, early organogenesis, 2 replicate embryos per stage. So here you see 1 embryo imaged from the dorsal side, 1 imaged from the ventral side. They're highly reproducible. I hope you can appreciate the large diversity of gene expression pattern in our data. They may look like 500 in situ hybridization patterns, but bear in mind that here every single cell has 500 genes probed in them simultaneously, and if you zoom in, you can even see the subcellular distribution of the transcripts. So in the lab, we also have single-cell multi-omics data, which means single-cell RNA-seq data that characterize the transcriptome, and single-cell ATAC-seq data that characterize in the same cell DNA accessibility patterns. Using a computational method called tangram, we can infer where these single cells come from and impute the rest of the transcriptome as well as nearly 300,000 DNA accessibility patterns. So we've made this into an online resource in an interactive web browser consisting of measured, imputed genes and imputed ATAC peaks. So if you work on zebrafish or early embryos, I highly recommend you to check it out. So from here, I will switch gear to talk about how we mine this data set to learn how we answer biological questions. We actually have a lot of findings on analysis of a single stage embryo, including expression statistics, co-expression of genes. We can see temporal transitioning within the same stage. But today, for the sake of time, I'm highlighting 2 stories, as I promised at the beginning, how we connect the stages to put these gene expression patterns in motion. So the first question I want to answer is how do gene patterns change with cell dynamics? For this, we utilized an in total imaging data set that was acquired more than 10 years ago. And we try to use the fate map offered by these live imaging data sets to connect the stages. We've developed a computational framework called Merfish Fate, where we map the corresponding cells between the Merfish embryo and the live imaging embryo, track the cells um, from early gastrula to mid gastrula stage and map these cells back to our mid gastrula stage Merfish embryo. In this way, of course, it's not stereotyped, so it's not a cell by cell correspondence, but we create a region by region correspondence map of the corresponding cells linearly related. Um, so we can track the dynamic of gene expression. So we found a lot of interesting gene expression dynamics. For example, here are the expression patterns at early gastrula and mid gastrula. Here's the log fold change with red showing increasing and blue indicating decreasing. So you see that some genes are de novo expressed, induced, and some other genes are scalped away to form their final expression patterns. What is very interesting is that we found a lot of mixed regulation patterns. Here's an example of the gene called TBXTA. If you're familiar with mammalian development, this is the brachyury gene, which is a famous marker for mesoderm identity. So if you just look at the gene expression pattern, you see that it's at the margin of the early gastrula stage and it's at the margin of mid gastrula stage. You may say, oh, this is boring. The cells just passively stay there. But once we incorporate the cell dynamics, we found that the cells that are closest to the margin actually downregulate this gene as they involute, and the pattern we see actually de novo induced from cell tiers closer to the animal pool. Very interestingly, we see that the induction and suppression pattern of this gene correspond to other genes measured in this data set, suggesting this regulation might be modular. Finally, I want to highlight a story about tissue boundary formation because it's the result of gene expression and cell movement. So we know that during development, boundaries could be challenged by cell proliferation or noisy signaling environment. So there are two ways of forming a sharp boundary. Either you can switch the identity of cells to conform to their neighbors, or the cell fate could be pre-specified and the cells could move to the corresponding compartment through cell sorting. Which case is it in our early development? So we've identified a sharp boundary between the notochord and somatic mesoderm at the mid-gastrula stage. Here I'm showing you expressions of genes as a function of distance to the boundary. You can see as soon as you cross this boundary, you shift from one category of gene to a second category of genes. Interestingly, when we track back to the progenitor region in early gastrula stage, we found that except for a small set of genes that are expressed in the dorsal side, the majority of genes change their expression patterns dramatically during the boundary formation process. This indicates that it is cell state change that's mediating the boundary formation. So what about cell behavior? So we use slide sheet imaging again. So we followed behavior, the movement of all the cells, identify their fate based on a transgenic marker. So by the end of the movie, we know which are somatic mesoderm cells, which are notochord cells, and we track them back and analyze their behavior. To our surprise, this is an amazingly orderly process, where you see here I plot the distance of the cells to the boundary as a function of time and their identity. You see that anywhere during development, if I draw a line between the two cell populations, the red cells stay on one side of the boundary and blue cells stay on the other side of the boundary, and they rarely mix. So this further confirmed our conclusion that this formation of this sharp boundary is mediated through cell state change rather than cell sorting. So I want to do a quick summary of what I just presented. We Murphy measured spatial pattern of 495 genes at subcellular resolution, integration with single-cell multi-omics data generated in the atlas publicly available online. Connecting WeMurfish with live imaging fate map reviewed unexpected gene dynamics. Cell state changes rather than cell sorting generates sharp tissue boundary during gastrulation. Finally, I just want to emphasize that Murfish is not just developed for early embryos. It's a generally applicable method for thick or whole tissue spatial transcriptomics. We worked pretty hard to push the thickness limitation of the sample from 10 microns to about 300 microns. So if you're interested in using or adapting the technique, you're welcome to check it out. So finally, I just want to thank everyone involved in this project. It has been a fruitful collaboration between our lab and Bogdan Bintu's lab at UCSD. Jacob is a graduate student in our lab who and I co-developed the WeMurfish method, and KJ is an undergrad student in Bogdan's lab who developed the Murfish Eyes website. Thank you for your attention. And with that, I'm happy to take questions.
Thank you very much, Yinan. Again, please ask your questions using the Q&A box. And while you're thinking, I'll start with a couple of questions. My first question is, for cells that have very complex morphology like neurons, how easy is it to tell which transcripts are part of which cell? Do you also get some membrane information that you can use to sort of characterize which transcripts are in particular cells?
Yeah, that's a very good question. Actually, we use this cell pulse and machine learning based cell segmentation method, and we only use the cell membrane data. Of
Hmm.
course, for cells with complex morphology like neurons, I mean, luckily, even at the latest stage, the neurons have very short processes, so it's not a big problem. We did so we did try a couple of other methods. For example, there are ways where you can input the location of the transcripts, and based on co-expression of genes in in the superpixels, it can resegment your data based on the similarity of. Unfortunately, we're not super happy with the results because it gives us too many small segments.
Right, yeah.
So it's just so we tried our best to segment the cells and while reporting what we believe is the actual real number of the cells in a data
Mhm.
set. Yeah.
Great, thank you for your answer. And my second question is, you look at around 500 genes
Yeah.
simultaneously. Is there a limit to how many you could look at depending on the amount of rounds you can do of the sort of probe and removal of the probe?
Actually, we haven't reached the limit yet. So it's the trick is that we really translated this imaging of the RNA space into DNA space by conjugating these DNA molecules into the acrylamide gel. So this makes the sample very stable for months. So right now we're doing 40 rounds of hybridization. So I could say we can easily double it. It's a matter of time. And the other thing we haven't really optimized is the density of the code book. Right now we are really on the sparse side. We are a bit afraid of molecular crowding, so we didn't put as many genes into the same hybridization rounds as like the commercial Merfish does. So by doing this, like denser code book combined with longer imaging, you can definitely go longer. But what I want to emphasize is our method is very biologist friendly in the sense that our code book is not hard coded in the library. So we design in a way that you can, for example, design a library of 10,000 genes and you can selectively image. You can say, I just want to image three or five or dozens of them. You don't have to redesign your library. They don't have to be imaged as a set. So this offers very flexible. So you can just do sequential one gene at a time, or you can do a set of 500 or 1,000. That's your choice.
Great, thank you. Very cool. Any questions from people? Maybe I can. Oh, we do have a question in the Q&A box. So Ruth Stifles is asking, oh, Osvaldo, a question. Osvaldo is still here. Um, if you've perceived any cytotoxicity when using EDU.
Uh,
Ah, yes, we've done it. And we actually touch a little bit of that in our manuscript. So we would normally recommend 1 to 3 hours of pulse chase labeling, but I'm conscious that a lot of people do several days, including such as when they study embryo development in low proliferative tissues such as the heart. So that development paper actually used a lot of hours of sort of from pulse to chase detection. Um, that actually stems some of the body of work that I'm developing right now. So I'm actually working very actively in understanding ADU and thymine analogs cytotoxicity, and I'm understanding that from a very in-depth point of view. I'm sort of using multi-omics approaches, panomics really, from epigenome, you name it, kind of run on ATAC-seq, single cell RNA sequencing in different models to understand DNA replication stress. And we're actually using EDU as a mimic of DNA replication stress. And we're comparing the effects of EDU with actually acetylcholine and hydroxyurea. So it is cytotoxic. And we have, for example, beautiful work right now in which we have somehow show hematopoietic stem cell depletion using long and repetitive injections of EDU into animals, which is work that a lot of people do, particularly in cardiovascular research and in other models such as cancer as well. So yeah, that's something that we're really looking into it right now as a model of detecting proliferative cells, but also as a model of understanding cytotoxicity and mechanisms of replication stress. Sorry for the long answer.
Great, thank you very much. So I'll finish with one last question for Yinan.
Mhm.
Um, so you mentioned that this can be applied to other thick tissues. Have you looked beyond zebrafish at other model organisms? Are there any that are particularly tricky to use with this method?
I mean, so far in the lab, we have been applying it to like the zebrafish brain
Hmm.
and killifish and some human gastroloid
Okay.
samples. So looks like the probe design um is quite is working for different kind of genomes. So depending on the density of the tissues, you may have to tweak how you cast the gel a little bit to avoid like squishing the sample.
Hmm.
But beyond that, we are quite happy with the result.
Great, thank you. Thank you very much to both our
Alex,
speakers. Oh,
can I ask something quickly
Mhm.
to Jinan?
yes, go for it. Yes, we have time.
Are you looking for collaborations? And just because we're developing some body of work about, how we affect gastrulation in silverfish, and particularly looking at the heart and using, a variety of cytotoxicity elements, or are you pretty close in general? I mean, we don't really want to develop this in-house because I know that it would take some time, but how open are you for collaborations, guys, in general?
Yeah, so we are very happy. I mean, we are ha very happy to share the protocol. I mean, it is actually uh, already with the bioarchive version, you can follow a lot of it.
Hmm.
So we are very open to share like our perfusion system, how to set it up in the lab. The only limitation is we may not have the capacity to support people bringing samples to our lab because we already have like four ongoing projects on our
Yeah,
microscope with four
yeah.
projects like running day
That's
and
kind of what
night.
I was thinking. Yeah, just to
Um,
create a perfusion system. And what
yeah.
light sheet imaging system are you using or have
So
you been
for
using?
the Weimerfish, it is spinning this confocal.
A spinning disk. And
Yeah,
yeah,
so
but
this allows single
okay,
molecule.
so
Like for the
spinning disk
live
is just one.
imaging, it was size microscope. Yeah,
Okay.
yeah,
Okay, yeah. We have them both. So
yeah.
lovely work. Thank you. Thank
Thank
you, then.
you.
We do have one last question, which will be a shame to miss. So Mega is asking how you reconstruct the transcriptomic data virtually uh, through the Weimer-fish.
So is this about how we incorporate the single cell multiomics data with Swimmerfish map? I understand that, or uh
I think you can, yes.
yes, okay. Yeah. So this is a deep learning based algorithm called Tangram. So it's trained on a shared set of genes, trying to use the existing gene to predict the genes that you haven't measured. So by learning this transformation matrix, so you basically estimate the probability of your single cells coming from the cells in your Murphy's data. So after learning this matrix, you can just multiply it by your real single cell data and get an expression map, the spatial map. I mean, similarly for the uh, DNA accessibility data, because it's multi-omics, so we have the accessibility score and transcriptome from the same cells. We just use the shared genes to estimate this.
Okay, great. Thank you very much.